Hello, we are Bio 2400 Genetics Group 9. My name is Justin Haoko.
I'm Ashley Mendoza.
I'm Sahil Gulshaw.
I'm Marianne Doggle.
I'm Jita Lawon.
And I'm Alexandra Gamboa.
And today we will be saving the bees for a better future.
My name is Jita Lawon, and I'll be talking about the goal of the project and the reason why we are saving the bees. The honeybees play a huge role in agriculture productivity, and they are a crucial pollinator. According to BBC, bees pollinate about 70% of the crop species that feeds 90% of the world. Therefore, the declining bee population can also threaten the existence of human beings. In recent years, the honeybee population has begun rapidly declining, with one reason being the consistent use of pesticides. Some pesticides are toxic to bees and will have a sublethal effect, which can result in reduced level survival, alter foraging behavior, and shorten lifespans. The use of pesticides is further facilitating the adverse effects the declining population has on the environment. Examples of the most common pesticides are pyrethroids, DDT, monooxygenase-mediated insecticides. One solution for saving the bees is to genetically modify them to become resistant to those pesticides. This can be done by taking the CYP4E2 gene from the fruit fly genome and amplifying it using PCR. The PCR product will be used to clone into the plasmid, which will then be inserted into the bee.
My name is Justin Haoko, and I will be talking about the PCR procedure to replicate the CYP4E2 gene. We will get the DDTR gene from the Drosophila melanogaster genome. We will use the PBAC6XP3 rubia plasmid that already has the DDTR gene separated from the rest of the Drosophila melanogaster genome. Polymerase chain reaction, or the PCR procedure, will be used to purify and amplify the DDTR gene from the plasmid. The product of the PCR procedure will be cloned into our plasmid. The first step of the polymerase chain reaction procedure is denaturation. Denaturation of DNA happens by heating plasmid to 98 degrees Celsius for about 15 to 30 seconds. And during this, the double-stranded DNA is denatured to single strands due to the breakage in weak hydrogen bonds. The second step is annealing. The reaction temperature is lowered to 50 to 60 degrees Celsius, and then annealing happens. This allows the primers to bind to their complementary sequence in the template DNA. The primers that are at the ends of the DDTR genes are mixed in with the denatured plasmid. The last step of PCR is extension, or also known as elongation. This extension happens around 72 degrees Celsius, and in this step, the polymerase enzyme sequentially adds bases to the 3' end of each primer, extending the DNA sequence in the 5' to 3' direction. Taq polymerases and DNTPs are added to the plasmids and primers to amplify the DDTR gene.
My name is Alexandra Gamboa, and I will be talking about the key procedure to cloning the DTR gene into the bee. A PCR reaction is an efficient way to amplify a sequence of interest, and then it is joined to the vector or a single base overhang ligation prior to the transformation. Early PCR cloning often use Taq DNA polymerase to amplify the gene, which was mentioned by Justin. The result in a PCR product is a single template independent base addition of a adenine A connects to the 3 end of the PCR product. Through the normal action of the polymerase, these A-tailed products are then ligated to a complementary T-tailed vector using a T4 DNA ligase, followed by the transformation process. DNA polymerase are constantly used to amplify DNA sequence, with the PCR product containing no 3' extensions. The end fragments are joined to a plasmid vector through a typical ligation reaction by the action of an activated vector that contains an attached enzyme, typically known as telomerase 1, which facilitates the vector to insert the joining. Some PCR cloning systems contains vectors that include a toxic gene into which the PCR products must be successfully ligated to allow producing of the strain that takes up the recombinant molecule during the transformation. In the plasmid, we'll be using the DTR gene that will allow bees to be resistant to the pesticides that are currently killing them today.
My name is Sahil Gulshaw, and I will be talking about the anticipated effects of the genetic modification that will be applied to the honeybee. Increasing the bee's resistance to pesticides will increase the survival rates of bees in areas in which pesticides are used, such as farms, and help to mitigate the negative effects that pesticides have on their population. If the CYP4E2 gene is successfully expressed by the bee, it will allow the production of DDTR or DDT resistance to take place, which in turn will allow the bees to metabolize and break down certain synthetic pesticides, such as pyrethroids and DDT. This would effectively reduce the fatality rate the pesticides have on the affected bees. This would also allow pesticides to still work effectively against their intended targets while not affecting the honeybees in the area. Successful modification will help reduce the rate of decline in their population and may even aid in eventually reversing the decline as well. The increase in the survival rate of bees in areas where pesticides are commonly used will lead to an increase in the overall population, in turn positively affecting the local environment. The increased rate of pollination due to the greater amount of bees would benefit both the agriculture and other flora in the area by facilitating the reproduction of plants.
My name is Mary Ann Doggle, and I will be talking about the genetic strategy on how to put our plasmid into bee zygotes. The genetic strategy that we will be using is the transformation procedure or the transfection procedure. To add on to what Alexandra was saying, we need to connect the DDTR gene and the plasmid together using DNA ligase. Afterwards, we will incubate bee zygotes and the plasmids together. To get the plasmid into the bee zygote, we will use selfectin or selfectin 2 reagent. This reagent transfects the plasmid into insect cells. It will take about 30 minutes for the entire process to occur. Then we will place the bee zygotes on a medium with nutrients for the zygotes to grow. In addition, antibiotics are added to the medium. The antibiotics are there to kill off any bee zygotes that don't take up the plasmid. However, the bee zygotes that do take up the plasmid will be resistant to the antibiotics because it has the antibiotic resistance gene. Therefore, they will grow on the medium. In addition, the plasmid contains DDTR gene, so the bee zygotes that take up the plasmid will have the ability to express the enzymes for pesticide resistance. The remaining surviving bee zygotes will eventually reach maturation, then that will be the end of the plasmid transfer.
My name is Ashley Mendoza, and I will be talking about the possible ethical issues that come with genetic modification. Issues to consider when genetically modifying organisms and reintroducing them into the environment include the survival and reproduction of the bees, since part of our goal is to increase the bee population. In order for this project to be successful, the new pesticide-resistant bees must be able to reproduce to ensure the new resistance will not be short-lived. One issue to consider could be how the pesticide-resistant bees will interact with other organisms and what effects they might have on the environment. There may be a chance that the bees can cause harm to non-specific targets or displace the native bees from the area. Environmental monitoring, which can be expensive, will be needed before and after release of the bees in order to manage any potential environmental risks and deal with any adverse effects. The process of removing the gene, amplifying, modifying, and inserting it into the bee zygotes will not harm existing bees, which will instill confidence and peace of mind to bee activists. If this project is successful, the pesticide-resistant bees can benefit other organisms and the agricultural industry.